your instructor, Jeevarathinam, Assistant Professor of Home Science, V.V. Banniya Perumal College for Women, Virudhunagar. In this course, Food Microbiology, you study about the microorganisms that is present in the food that causes food spoilage, that microorganisms involved in food preservation. Food Microbiology, in this first module, you will study about culture media. What is culture media? Humans need food, shelter and dresses are their basic needs. Like humans, microorganisms are also require some things like media. Media are food material or substances that contains all nutrients. The culture medium supply nutrients for the microorganisms to support and the survival of microorganism. The culture medium also called as growth media. Next one, what are the ingredients present in the culture media? The culture media contains peptone, beef extract, yeast extract and agar. In this, the beef extract, peptone, yeast extract are act as a carbon, nitrogen, amino acid sources. This agar is a solidifying agent. It is extracted from red algae. It mainly used in the food as a thickener. Here, the agar is used to prepare solid media. Next one, classification of culture media. The culture media are classified based on their physical state, nutritional factors and application or chemical composition. First one, the culture media classified based on their consistency, divided into three groups. One is liquid media, solid media and semi-solid media. First one, solid media. The solid media contains 1.5 to 2% of agar. So, it is in the solid state. Example, nutrient agar, mechaniki agar, blood agar. This medium contains 2% of agar. So, these type of mediums are used in the enumeration or counting of microorganisms. Next one, liquid media. This liquid media is also called as broth. It does not contain any agar. So, it is in the liquid form. Example, nutrient broth, tryptic broth are examples of liquid media. Next one, semi-solid media. This type of media contains only 0.5% of agar. So, it is in between the liquid and solid state. Example, this type of media are used in hanging test. That means hanging drop test means this type of media used to identify the motility of microorganisms. Next, based on their nutritional component, the media are classified into four types. First one is simple media. Simple media, it is a general purpose medium because it contains common ingredients only. It supports the growth of non-fastidious organism. Non-fastidious organism means that organism does not require any special supplement. Example, nutrient broth, peptone broth. Complex media. Complex media means the composition of this media are unknown. This media contains extracts of plant or animal tissues. Example, tryptic soy broth, blood agar and nutrient broth. Next one, synthetic media. Synthetic medium is a defined medium. So, the composition of this medium is very well known. It also purely made from chemical substances. Next one, the culture media are classified based on their application or chemical composition. First one is basal medium. It is a non-selective medium. It means it's a very simple medium. Example, nutrient agar, we can easily produce the nutrient agar in our lab. It is mainly used for non-fastidious bacteria. Enriched media. Enriched media means this type of media contains some special supplements. These supplements required for the growth of particular microorganism. Example, chocolate agar, blood agar. Then selective media. These type of media only grow the certain type of microorganisms. It incubate other microorganisms. The substances like bile salts, antibiotics, dyes, and also by adjusting pH, we can grow certain type of microorganisms. Then indicator or differential media. This type of microorganisms differentiate the various kinds of microorganisms. These media contains indicators like methyl red, neutral red, phenol red. It it shows the visual changes on the nutrient agar plates. Application of culture media. Culture media is a fundamental of microbiological test. It used in all microbiological test. First one, it is used to identify the cultural characteristics and morphological characteristics of microorganisms on agar plate, agar slant. Then nutrient broth. Nutrient broth mostly used in fermentation studies. It is the culture media also used to isolate or separate the pure cultures by streak plate, bore plate or spread plate. It also used to store or preserve the culture for long days. The culture media also used the biochemical reactions like fermentation, lactose fermentation and also starch hydrolysis. So, this culture media is a very important one. Without culture media, we can't produce the microorganisms in our laboratory. So, that's all about culture media. Thank you.